Hi, this is Miss Linton, and this is an excellent grouping of young men and women for our Honors Biology Unit 5 review. Say hi. Hi. All right, so I'm going to walk you through. These are the three big topics, if you don't remember. These are what you need to know. So I've set up some questions that I might pose to you on an exam so we could discuss them now, okay, in a safe environment. So the first one is, can you diagram the cell cycle and name the parts and what occurs? And if you're wondering to yourself, should I screenshot each one of these? Or you could just look in a folder I created in our group, in our documents that we share. I made a folder. I don't know why I wasn't doing this before, reviews. And I put this in there as a PDF for you. Okay? So can you diagram the cell cycle and name the parts and what occurs? Could you do that? Okay, what would you tell me the cell cycle consists of? What would you have me draw?
A circle.
That's what I would draw too. I would draw a circle. Then what would you draw? A slice of pie? Okay. How big of a pie? Like this, maybe? Okay. Kind of like a Pac-Man upside down with braces? Okay. And then you would say to me that this whole section right here is what? Interphase. And you would break interphase down into what? What would you call it? What else? And? And would you know what happened? What would you call this right here? M. And what would you divide that into? And mitosis. Good job. All right. Would you know what happens in each one of those stages? Yes. What would you tell me happens? What color have I not used? I don't know. What happens here? Increase in size. And what else do you make? Organelles. Good. What happens here? Synthesis of DNA. That was super clever. Okay, and then what happens in G2? Proteins. Good. All right, then could you break down the M stages? Yes. What are they? Prophase, metaphase, anaphase, telophase. Perfect. And you could tell me what happens in each one of those? Maybe we will sing a song right now? Yes. Okay, here we go. What's the first stage? Prophase. The chromosomes. Now, when they become visible, they're what? Two, because these are held together by a centromere. Okay, just a second. And these would be a small pair. Do we really care about that in mitosis? No, we care about that in meiosis. When we go from 2N to N. Do you still do the S stage in both mitosis and meiosis? Yes. Yes. Okay, here we go. The chromosomes become visible, the nuclear envelope and the nucleolus disappear, radiating microtubules, asterisks. They meet in the middle at metaphase. Single file, right? Because we're doing what? What are we doing? Mitosis, good. They meet in the middle at metaphase, away at anaphase. What goes away? Okay, two nuclei at telophase, cytokinesis. Muy bueno. All right, so it sounds like you know what's going everywhere. Let's look at this one. Could you diagram a cell and the stages of mitosis with a cell that had 3 chromosomes, with one that had 4 chromosomes, with one that had 5 chromosomes? Could you? You did this with licorice, my friends. Okay, should we practice? Okay, let's do it with just 2 chromosomes, all right? So I would probably show a stage. What stage would this be right here? Okay, perfect, good. All right, and then I'd be going into mitosis. What's the first stage of mitosis? Prophase. How could we draw? What happens during prophase? Chromosomes become visible, right? Here, let me get some other color here. What else happens? What does? Nuclear envelope starts to what? Disappear. Asters form. What else do you have? Yeah, microtubules are starting, right? Okay, but do you see how I can clearly see that there are 2 chromosomes? How would I write that mathematically? 2 chromosomes. 2n equals what? 2. This is a cell where 2n equals 2. Do you agree with me on that? If I had 3 chromosomes in there, which would be an odd number, that would be 2n equals 3. If I had 4 chromosomes in there, it would be 2n equals 4. Right? That was your licorice. Right? Okay then. All right. What comes after prophase? Metaphase. So how would it look? Single file. What else would I see? Yes, I would. Something like this, right? Do you agree with that? Yeah. Okay, what's the next stage? What would that look like? Sister chromatids. Okay, then what? Telophase. And I would want to make it distinct so I knew which chromosomes were in there. I would not make solid dots. I would not make like sunglasses. See what I'm saying? I would know that I would want to see that there was a blue chromosome in each one and a red chromosome in each one. Do you see what I'm saying? Then I would do cytokinesis. And you could make it a solid thing. But make sure I can see in telophase that you have separated those sister chromatids. Yes?
Do you ever need to label like proteins and stuff?
No. It would be very just general if I asked you that. If. Okay, more questions on that? You feel good? So we would know how to do it. I asked you if you had 3 chromosomes, if you had 4 chromosomes, if you had 5 chromosomes, right? I'm not asking you to do meiosis, right? So if I was asking you to do meiosis, I'd probably do 2, 4, 6, 8, right? I'd do something like that, because then you would line up with your homologous pair. And if I'm just doing mitosis, I could have any number. All right, moving on. Can you name and describe 3 different ways meiosis contributes to genetic variety? Tell me now.
Independent assortment.
Okay. Now you know what independent assortment is, right? Because we've talked about it in genetics. The homologous pairs can line up any big way. So, for instance, I could get a big T with a big R. Let's say I'm tall and round, and I would tell you what would be another thing I could probably get? Big T with a little r, and those would all be what? Gametes or genotypes? How do you know that? There's only one member of the homologous pair. There's only one kind of T, right? If I said to you this was the parent, that could be a genotype, right? Because I have my homologous pairs. Could this be a genotype as well? Yes, because I have 2 T's and 2 R's. Yeah? Okay. All right, independent assortment was one way. What was another way? Crossing over. By the way, let's talk about when that happens. When does this happen? When? Metaphase 1. When does this happen? Okay. Now, those are all during the process of making gametes, right? But there's one other reason. Because we do meiosis and we're down to haploid cells, what can happen? Fertilization. Yes. Fertilization. The joining of gametes, right? Comfy? Questions? Shadow? Sunshine? Sunshine? Okay. Can you describe the similarities and differences between mitosis and meiosis? Could you make a chart? How are they different? How are they the same? Could you compare meiosis 1 and meiosis 2? Could you compare cytokinesis in plants and animals? I'll let you do that. You want to make sure you're clear in your mind, like when does everything happen, right? Mitosis always goes, mitosis, the chromosome number stays the what? Yeah, then the number is the same. And how many cells do you make when you do mitosis? 2 cells. Whereas in meiosis, what do? Come on.
4 cells. We make 4 cells.
Okay, I agree. 4 haploid cells. What's another way of writing that mathematically? Right? In mitosis, most of the time when you're doing mitosis, you're going to say 2n goes to 2n, but I'm going to also tell you n could also go to n. That happens in plants and alternation of generations, so I don't want to mislead you and say that can't ever happen. But whenever the 2n goes to the exact same 2n, which is usually what you're going to be doing mitosis for, that's all. You don't need to worry about this. We're not talking about that here. 2n to 2n, that's mitosis. Meiosis, 2n down to n. Usually 4 cells, where this is making 2 cells. What could you tell me if I was comparing the parent cell to the offspring cells? What could you tell me about mitosis and meiosis if I was going to compare and contrast them? Yes?
In mitosis, the parent cell is going to be like the exact replica of the daughter cell.
So the daughter cell is a replica of the parent cell. Good. What could you tell me about meiosis and those offspring cells? Yes?
Those are going to be different.
Yeah, they're always going to be different, right? Because you have a mixing of the maternal and paternal, you know, when you make your gametes, however you make them, okay? It's a mixing of your maternal and paternal, and you generate that through independent assortment and crossing over, right? All of those. Okay. Could you tell me the difference between meiosis 1 and meiosis 2? Meiosis 1, what? Separate amongst pairs. Meiosis 2? Cytokinesis in plants and animals. Tell me what you have where? Plants, they make a cell plate, which becomes a wall. Because a plate is thinner than a wall, right? Unless it's a wall that you have to look through. Or unless the river or the body of water becomes part of the wall. It's all about border security. What could you tell me about animal cells? What could you tell me about animal cells? Cleavage for us, right? Animals have cleavage, not plants. You'll remember that, right? All right. Cleavage, cleavage, cleavage. Is that what you're doing right there? Okay. People don't know us, and they might be listening to this. Okay. Can you describe the similarities and differences between oogenesis and spermatogenesis? What are we doing? Oogenesis. 1 large egg, 3 little polar bodies. But in spermatogenesis, you'll make 4 haploid sperm. What does he wear on his head? Acrosome, his little cap. What's inside his cap? To digest when he wants to make a diploid zygote. Remember? All right. Okay, good with 5? Okay, while you're being chatty Cathy, still be stressed. Some of you are like, I can do this, so I'm going to start laughing and talking to my friend. No. Okay, we need to stay focused all the way through. So chapter 22 was development. Remember, that was a partial. We didn't do all of it. Can you name the structures of fertilization? Shall we do them now? Here comes Mr. Happy Sperm. He's got a cap called? What's inside of it? Enzymes to digest the zona lucida. And we form a diploid zygote. That's the zygote, sperm, egg, zygote. And you're going to do what? Cleavage, cleavage, cleavage to form a solid ball of cells called a morula. And then we're going to pump. What are we pumping? Where? Inside, right? And what's going to happen? Water will follow and we'll form a blastula. And a blastula is a hollow ball of cells. What day do we form it on? Day 5. Good. All right. And now we're ready to form a gastrula, because it looks like we're deflating. And at first there's just 2 layers in our gastrula, but ultimately there's going to be 3 layers. Let's talk about those 3 layers. What's the outermost layer going to be? And what will it be? Nervous system, skin, lens of eye. Okay, what's the innermost layer? What will that be?
Respiratory
Digestive tract and associated organs. What's the opening to the gastrula called? Starts Okay. So the space is called the archenteron, but the opening is called the blastopore. And it's either going to be your mouth or your anus. Good. So anus. It's going to be anus. Okay, and then after, and what's the middle layer? And what will it become? Muscle, bone, everything else, right? Okay. Okay, so where gastrula is any kind of layered ball of cells. Could have 2 layers, could have 3. Then we form a neurula, remember? And this is when what's the outermost layer called? Folds in. The process of folding in is called, the process moving around and folding is called morphogenesis. But when your, starts with an N, notochord from your mesoderm tells the ectoderm, that's induction. And that's peer pressure. One tissue influences another. Notice it goes cell, zygote, right? Then we go to, that would be a cell. What follows cells? Tissues. You get to the tissue level, and then you get to the organ, and then organ systems, and then organism. Just how we learned it. That's how you develop as well, in that same pattern. Okay, did we hit them all? I think we did. Can you name and describe all the stages of, we did. We just did. Why did I say this? The structures, the egg and the sperm. We did the structures, and now we did the stages. Questions, concerns? Okay. Can you name and describe the layers of the gastrula? Did we do that? Yes, totally did it. Check. Can you differentiate and give an example of induction? Yes, we just did. All right. Can you describe how mitosis and meiosis contribute to the stages of fertilization and development? What would you tell me? Meiosis makes the gametes, which get together through fertilization to form a zygote. And then what do they do from then on? Mitosis. Does that make sense?
Yes.
Meiosis makes the gametes get together through fertilization. They form a zygote, and then they're going to do mitosis. Good job. All right, chapter 23. You ready? Where are all these questions?
In your book.
In my book. No. I put them in on my website where you get all your group shared notes. There's a folder that says reviews. It's all in there. Yes. You can't find them anywhere? Did I put them in the wrong spot? Here, let me do it right now. I'll re-put it in there. It's not in group shared notes. It's in a folder called reviews. It's called reviews. It's in their group shared notes, labs, reviews. See it? Group shared notes, labs, reviews. You got it? Look for it so I know. Not there? You do see it? Okay, here, I'm going to put it there right now. Save as PDF on my desktop then. Save. Okay, sorry, delay of game because you can't find this. Make sure I'm going to the right folder. Okay, right now the first thing I'm going to ask you to do is make sure you understand Mendel's 3 laws, be able to name them, and give a concrete example to explain them. Can you do that? Name Mendel's 3 laws, name them, give a concrete example to explain them. Oh, you're right, it's not here. Weirdness. So sorry. Maybe that's why. Maybe I was not in it myself. goodness, I just saved that. Where did it go? Okay, I promise you I'll put them in. I don't know why it is struggling. Oh, I know I did. Okay. Desktop.
Yeah, all possible combinations. It's like
Now it's in there? No, I just made the folder. There it is. Now it should be in there. Refresh it, it'll be there. Sorry, it was because I had it untitled. I didn't recognize it. My bad. Okay, come back to me. Question number 1. Let's name the 3 laws. Law of? Give me an example. Tall and short. But what would be evidence of that? That would say if you had this genotype or this genotype, you would be tall. The only way to be short is if you have what? And no, you need to know the difference between genotypes and phenotypes. Yes? Okay, what's the second law? Law of segregation, which means what? Okay, so you separate them all against pairs. So you would separate, big T would be in one gamete. Let's say if you had a heterozygote, big T would be in one gamete and what? Little t in the other gamete. Okay, what's the third law? Independent assortment. And that would mean, like we talked before, big T with a big R or big T with a little r, etc. Okay, and you would give me examples of that. Another example that you could give me that's specific is you could talk about hair color and eye color, because then you could bring in what? Crossing over. Hair color and eye color tend to travel together. The only way to break up gene linkage is through crossing over. Good job. All right, next. Can you differentiate between a somatic cell and a gamete? First of all, what's a gamete? Sperm, sex cell, sperm or egg. Everything else is a somatic cell. Phenotype and genotype? Yeah, actual alleles. Phenotype is a description. Phenotype will always be a what? Word. Phenotype will always be a word. Genotype will be the actual genes. Homo heterozygous? Yeah, easy peasy. Blood types and multiple alleles. Blood types give us an opportunity to talk about 2 things. What 2 things can blood types give us the example to talk about? Multiple alleles and what else? Codominance. Multiple alleles because we have big IA, big IB, and what? Little I. Codominance in this situation, right? AB blood. Both the A is expressed and the B is expressed. All right? Incomplete. What's an example of incomplete? Yeah, red with white makes what? Pink. So what you want to really use there is to talk about how the heterozygote has its own unique phenotype. Right? Heterozygote has its own unique phenotype. Okay, what's another example besides red, white, pink? Yeah, curlies, 2 curlies making, yeah, oh no, curly and straight making wavy. Yeah. All right, polygenic. How is polygenic different than multiple alleles? How is polygenic different than multiple alleles? What?
It's guided by
2 or more sets of alleles. So you could have an R1, R2, a big Q, a little q, big L, little l. You could have all of these things polygenic, many genes, R's, P's, Q's, M's, and O's. Polygenic. And there you tend to see that bell-shaped curve. And all you have to do is look at our height, look at our skin color. Those are all polygenic traits. And those can be easily influenced by the environment as well. Questions, concerns on any of that? You want to be able to name and give a specific example. Describe, name, and give a specific example of these, because it's possible I will ask you that. Okay. All right. Like here. This one I either skipped it on the last one. What's the only one we didn't talk about? Sex-linked. Okay? So on sex-linked, we always need to consider that the females, right, have 2 shots at it, and the males only have what? 1. So this is what breaks the law of dominance, because it doesn't matter if he has a dominant or recessive trait, he's getting it. Whereas we're used to, in homologous, in diploid organisms like ourselves, we have to get 2 recessives before we express that trait, right? What's a really good example of sex-linked traits? Color blindness. What's an example of a recessive trait? No, 6 fingers. Oh, 6 fingers is dominant trait.
Yeah, so 5 fingers.
Oh, 5 fingers. I'm sorry, I'm with you. What else? What were some diseases we talked about with recessive? And what else?
Sickle cell.
What else starts with a T? Tay-Sachs. What was an example of an autosomal dominant disease? Neurofibromatosis. What was another one? Huntington's. Yeah, good. All right. Can you do a Punnett Square with 1 trait and give genotype and phenotype results? How about with 2 or 3 traits? Yes. Want to try one or no?
Yes.
Okay, can you cross for me? No. And I want to know, what are the odds of this? So in my mind, I'm crossing the T's, and I know every single time this one's going to give a little t, and this one what? Half the time it'll give the big T, and half the time it'll give the... So half the time I'll be big T, little t, and half the time I'll be little t, little t. Because this time, this guy's always going to give me a little t. So he's like, don't even consider him, right? So I'll get half the time little t, little t. Okay, now let's look at the R's. If I cross just the R's, what are the odds? You can see that Punnett in your head. What are the odds that I'm going to get little r, little r? 1/4. Do you see that Punnett, right? What was that, a little? 1/4. And then I look at the Q's. Well, half the time this guy's going to give a little Q no matter what, right? He's going to, and so he's always going to give a Q, little Q, but half the time he'll give a big Q, half the time he'll give a little Q, so I know this is half if I do the Punnett Square. So 1 times 1 times 1 is 1, 1, 2 times 4 is 8, times 2 is 16. Chance has no memory. So the next time it'll be 1/16 of a chance too of that turning up. If they happened in sequence, 1/16 times 1/16. Yes?
So you don't really have to do a Punnett Square because you can
No.
just see that 50% of the time it's
Yep.
gonna give that team.
Yeah, I probably would do a Punnett just to be sure because I'm a little OCD. But doing this right now with you, I'm just like, nah, half the time.
And can you also do foiling to
Sure.
be able to do that instead of a Punnett?
Yeah. Well, yeah, I don't know. I say foiling for forming gametes. Yeah. All right. Next, can you explain why a test cross is necessary? Who would I need to test? What kind of individuals do I need to test? If they have a dominant trait and I don't know their genotype. I know their phenotype, it's dominant, right? So who do I test them with? Who do I test them with? Homozygous recessive, because I know they're always going to give the like recessive trait every single time, and then I look to see what happens. If half the time it turns out big R, little r, and half the time little r, then I know this guy was probably hetero, right? But if every single time it turns out round, he was probably homozygous dominant. Okay? Do the practice problems in the chapter, and at the end of the chapter, answers are in the back of the book, and you need to do your additional objectives. And I forgot to put this in your thing. Let me open up. Where do you see our last chapter? There it is. Let's look at some pedigrees, because I'm telling you right now, pedigrees will be on your test. Okay? So you just want to feel comfortable with them. I have a tech light meeting today. Any too? I hope not. Okay, so let's look at some pedigrees. Let's look at this one. Okay? Let's decide what's going on here. How could this be inherited? What's going on? Would this work for a homozygous dominant trait? Okay, let's just write it out. If this is a dominant trait, let's use letter A's. Okay, if this is a dominant trait, that means they have to have, and that's what you always want to start out with. Oh, if the trait is dominant, and then work your way back and see, will it work, and could you figure out the parentage? So this is a dominant trait, then this is a dominant trait, yes? They don't have whatever this is, so they would be what? Little a, little a. Okay, I know that if they have two little a's, then their daughter here is always going to get a little a, because that's all. We're not talking sex linked, okay, so don't take that out of your mind. Don't let that confuse you. Every single time, this dad is always going to give a little a. I know this individual has a big A because we said it's an autosomal dominant trait, right? So it's big A. Do I know what this letter is right here? No. I have no idea. So will this work for an autosomal dominant trait? Yeah. Is it possible? Yes. Yeah. Okay, so and then we just worked our way through. Now let's try a recessive trait. Let's see if this will work with a recessive trait. If it's recessive, then what would this be right here? And what would this be? Okay, so we know one of these little a's could come here. This little a would have to come from here. Could that happen? If this was a big A. We're just trying out different traits. We're just like shoes you're trying on, right? Here's an inheritance pattern. Will this shoe fit? Will this shoe fit? Is it Cinderella? Is there only one way? Looks right now there's two shoes that fit this foot, right? Now let's try the sex-linked shoe. If this one is a girl who has it, what would she have to be? Yeah? Okay? And the mom is what? So we know where one of these comes from. Where would the other one have to come from? Here. Does he have the disease? No. So he's got to be X big A Y. So could this happen? No. Could this have happened if she were a he? Ooh. Could it have happened if she were a he? Yes, it could have happened if she were a he. Because then this could be a what? Y that you got from the dad, and this little X right here could have come from mom, yeah? Okay, let's try another one. Yes?
The mom doesn't have to be little a, little a.
She does because she's colored in, and we said it was a sex linked.
Okay, yeah.
She wouldn't have to be, but she is because we're saying it's a sex linked trait. Okay, so let's try all those three shoes on this one. Do all three shoes fit on this one? Should we start with the recessive one first? Okay, so if we say it's a recessive trait, we're going to right away make this what? Little a, little a. Okay, here are the parents. Could they give little a's? What would they be? So does it work with recessive? Yes, that shoe fits. Let's try on another shoe. If we make this a dominant trait, what would this be? big A, I don't care, right? Can I get that from either one of them? No, because if I say it's a dominant trait, then these have to be, yeah, it doesn't, I mean, one of them would have to be and they're not because they don't have the disease. So does that shoe fit? No. Okay, let's try the sex-linked shoe. Okay, he has it. What would we do? Little a, he has the disease, and I'll only give you recessive ones. And then what? Y. Okay, dad doesn't have it, so what would he be? So that much works because he just has to give the Y, right? Okay, could we make it so the mom does this? What would it be? So does the sex-linked shoe work? Yes. Okay. So let's try one more. What do you think? What shoes can fit? Would sex-linked fit?
Yes.
Okay, and if I just said sex-linked, I would go X little a Y, and I would go dad doesn't have it, so that could be X A Y. That works. Mom could have given it. X big A, X little a. Who could she have gotten it from? Oh, her dad. Who has it? Right? See how I did that? So yes, the sex-linked shoe fits. Does the recessive fit? Yeah, right? Would that work? Yeah. Would the dominant one work? What? Would it? No. Okay, because if this person had it, they would have to get it from someone. If you want to grab all three on one paper, here they are, if you would like that. It's not so much that you have to always figure out, like, and be the historian, but I want to see that you can think through all possibilities. Maybe I give you a pedigree and I said, for instance, let's say this one. What if I said this was Al and this was Betty, and they're going to have a child. What are the odds that another child, they have 2 children, they want to have a third. What are the odds that their next child will have the disease? Well, let's figure out Al and Betty's genotype. Let's go with, what do you want? Which way do you want to go? Name a disease. I'll take it out. Sex-linked, Tay-Sachs, or recessive disease? Okay. So if this boy has that recessive disease, he's little a, right? What does that tell you about Al and Betty? Big A, little a. Do you agree with that? So if they were to have another child, it doesn't matter, boy, girl, because this is just a sex, oh, it is, never mind. No, we didn't make it, we just said recessive disease, that's right. It's a recessive disease, so it doesn't matter if it's a boy or a girl, it's all good, love them either way. So what are the odds that this child would also get Tay-Sachs? 1/4, 25%, right? Because if you do this Punnett Square, right, they'd have a 25% missing it all together, 50% of being a carrier, and a 25% of getting Tay-Sachs. You understand?
Yeah.
You need me to do another one, or are you good? Okay. Good job, you guys. I'm super proud of you.
Thank you, Miss Lily.
You're welcome. Don't stay up too late. Have a piece of toast.